I've been really like excited by the pace of progress in like LLMs and the potential of integrating them with scientific workflows to essentially empower the scientists doing the experiment, right? Um, will we be able to completely cut out the computational person? I mean, maybe, again, like maybe for some workflows we can, and maybe for the more complex stuff you need really experts. But I think the potential is there.
Hi, everyone. Welcome to episode 25 already. And here today with Chaitanya Joshi. Thank you so much.
Yeah, I'm based in Singapore these days. Yeah.
Like a little bit of an introduction.
Yeah, I mean, so we can briefly describe the central dogma of molecular biology, which is always a good context. It says that, in the cell and in on living organisms, the DNA carries these instructions, this genetic code, um, which gets converted to RNAs, mRNAs. These are called messenger RNAs. And then the cellular machinery converts these mRNAs into proteins. And the proteins go and do everything in the body. And there's this separation of DNA as this permanent storage, proteins as this functional molecule, and RNA or mRNA as this transient intermediary, this messenger, right? Um, and this is a basic picture. We learn about it in high school, but um, quite recently, like scientifically, quite recently, in the early 1980s, scientists discovered catalytic RNAs, ribozymes. These are RNA enzymes, so RNA molecules which are not transiently coding for proteins. They're actually carrying out some function similar to proteins. Um, and this discovery was really quite fundamental. It, shifted the perception of what the RNAs do, right? It it now suggested that RNAs are not just coding for information like DNAs and mRNAs. They code for a protein, but that they're capable of performing some function. And hence they're capable of forming some structures and performing some dynamic activities in the cell. And this really catalyzed, new research in biology, in RNA biology, leading up to, discoveries such as how the ribosome functions, for instance. The ribosome is the cellular machinery for converting mRNAs to proteins. So the protein factory of the cell. For a long time, it was thought to be a protein-powered machine, but as things advance, we now learn that the ribosome is actually an RNA machine. So the RNAs are performing the function there. Similarly, we started discovering a lot of microRNAs. These are small RNAs which are not coding for proteins. They may be floating around in the cell and they interact with mRNAs and they regulate how the proteins are produced. So this discovery of the ribosome really turned this central dogma on its head. And now, fast forward to today, RNAs are like this superstar of molecular biology, right? And, the RNA therapeutics are on the rise. And in terms of basic biological science, every few years, Nobel Prizes are being awarded for new RNA-related discoveries. So we're learning more and more about the roles of RNA, and I think, they essentially are involved in lots of cellular biological processes. The simple understanding is that they code for proteins, but they also do so much more in terms of protein production, regulation, and other, other sort of biological processes in the cell. So that's why it's, an exciting time to be in RNA biology, I would say. Origins of life research, right? One aspect of it is thinking about like what were the basic chemical building blocks um, available to us, right? The the other is thinking about evolution, right? So like nothing in biology makes sense except in the light of evolution, right? What was the molecular basis of evolution? What was the what is a biopolymer that is capable of kickstarting the evolutionary process? And based on what we know about ribozymes and what you just said, RNA is the most likely candidate to be capable of both carrying genetic information, so coding for like some genome, as well as performing some function. In this case, the function is self-replication or replication of other RNAs. That's how evolution would have kickstarted, right? We may have had something known as a primordial soup. So just nucleotides or like various atoms floating around, and these assemble together into a polymer, which by chance is now capable of recursively copying itself with some error, of course, right? And that's what people think led to evolution and hence life. And, this sort of smoking gun, or let's say like one argument for this is the ribosome. The ribosome is, as I said, like this very special machinery, right? It's a huge complex consisting of RNAs and proteins, which are actually very special structurally. Like they've co-evolved to like hold together this large machinery and perform its function. But the catalytic core, the functional part of the ribosome, is an RNA machine. And this suggests that, the initial function might have been performed by an RNA only machine, which then evolved these proteins to perhaps improve or optimize its function. And so, this is one theory for the origins of life, that it was an RNA molecule or an RNA like polymer which learned to self replicate and kickstart this evolutionary process, which eventually led to cells and humans and so on, right?
Yeah,
But that
right.
it must have started at a molecular level.
Yeah, I mean, I think it's super interesting, right? Maybe to even speculate on what possibly could have been, the sequence of steps that went from this RNA polymer, right, that somehow can copy or kind of catalyze formation of bonds that kind of create other RNA polymers, right, and then suddenly it starts developing into something more and more complicated. And that maybe DNA kind of emerged as a way to make the transmission of information more reliable, right? Because my understanding is that DNA is just much better, much more reliable at that as means of transmitting data, right?
Yeah. Yeah.
Um, so maybe moving a little bit beyond that, right? So maybe part of the reason why RNA is very interesting is because these molecules are very flexible, right? They can take all sorts of different shapes. Like, do we know a lot of RNA structures? I guess I can kind of guess the answer, right? But like, do we know a lot of experimental structures or not? If not, why are they complicated, right? And is there like I don't know, like a equivalent of PDB for RNA or RNA structures in the PDB?
Yeah.
Can you tell us a little bit about that?
Yeah. Yeah. I think like the situation there's a bit dire. Um, and that has been the main limiting factor for machine learning for RNA. So there are about 200,000 protein entities in the PDB, and there's about 15,000 which contain RNA. Most of these are from protein RNA complexes. So the protein is still the dominant part. Um, so at least 10x less data, which is also significantly less diverse data, because historically a lot of people did structural studies on very specific classes of RNAs, ribosomes, tRNAs. These are very well represented in the PTB, but more like more rare classes of RNAs, which we might be interested in designing or engineering, are less represented. Why is that, right? Um, I think I think like I think it one of the sort of easy reasons to give is that, hey, RNA is more unstructured, it's more flexible, it's more dynamic, and that makes it harder to crystallize or to um, view in cryo-EM and to obtain like these static snapshots of. I think that's like largely true. That I think basically like the experimental techniques need to advance to like be able to capture these RNA structures better. And there have been, in recent years, significant advances in RNA cryo-EM as well. So there's this hope that in the next few years, we sort of get an exponential speed up in number of RNA entities in the PDB. And yeah, I think there are other interesting reasons as well, right? I think that, a lot of times RNAs might be flexible such that, they form structures in presence of some partner molecule that, it is responsible for a function. And perhaps like just trying to, get them to form structures without that partner is not as biologically meaningful. This could be one of the reasons. I think other reasons could also be like historical interest, right? So there's like more interest in protein structural studies, and the sort of studies of RNA are relatively like fewer, right? But that's starting to change as well.
Yeah, of course. I mean, I can imagine, right? Like if you start from this preconceived notion that the RNA is just the the blueprint for a building, right? And then the proteins are the buildings. You study the building, you don't study the blueprint, right? Like the blueprint is just what you FedEx back and forth, right? But what matters is the building. And of course, that's, I guess we know now, right? That is not necessarily true. I wonder if with proteins, one of the things that happens is they kind of assemble into these tertiary structures because some amino acids like water, some others don't like water, and they kind of form like blobs, different structures depending on how much they like or they don't like water, whether they have charges and so on and so forth. But I guess we don't have that with RNA, right? Like there's a little bit less maybe um, tendency to aggregate in those specific ways, so that RNA can be more flexible and form all sorts of weird structures that maybe we just don't understand yet.
Oh yeah. So what's the structural basis of what's the mechanistic basis of RNA structure, right? It's fun to contrast this with proteins because people mostly know about proteins. So mostly the protein backbone is not charged, right? And some of the amino acids are charged and hydrophilic or hydrophobic. The RNA backbone of the polymer is actually negatively charged. Um, so for this entity to even start forming structures, you need like salts, you need these counter ions to balance the charge. And thus, under different concentrations, your RNAs may already fold differently, different concentration of the salts. Um, but so, what's the mechanistic basis of this folding? There are like mainly like two or like two main sort of drivers of this structure. One is hydrogen bonding to form what's called base pairs. So here, you have the backbone of the RNA or the DNA as well, and you have these bases, so these kind of these nitrogen-containing rings sort of sticking out of it. And what happens during sort of folding is that two strands go anti-parallel. So maybe imagine it like that, right? Anti-parallel strands whose bases stick out, and then they form hydrogen bonds to kind of hold the thing together. So it's really like a ribbon which is held together by some kind of crossings. Um, this is one. This is well known, and like this is also related to the genetic code that like um, A's and U's base pair and G's and C's base pair, and then in DNA A and T base pairs. The other sort of driver of structure, which actually like recently people think is even more important, is called base stacking. So you have these bases sticking out, and each of them contain rings. So I might get this wrong, but like in chemistry, there's this concept of pi stacking, right? So the rings from the bases, how they align with each other in 3D, um, can can lead to a very stable arrangement, or more or less stable arrangements. And this kind of stacking of the rings along the chain is also driving a lot of structure formation in these RNAs.
Yeah.
So different kind of forces are essentially at play in proteins and RNA, right? Uh, yeah, this kind of makes it interesting.
I'm not sure I'm a great person to explain, pi stacking either, right? But I guess like we could think of as a quantum effect that happens, right, when you have these different rings, right, that they tend to align with each other, right, instead of uh, kind of be out of alignment. But so we have those forces that determine the RNA structure, right, and then we know, you mentioned before, that it's been complicated so far to get some of these structures. So then I imagine the problem of even predicting the structures or designing structures would be even more complicated, right? Like, if we don't have many examples, if we can't really predict the structure from the sequence very clearly, how can we design an RNA for a specific structure that may be interesting for us?
Yeah, I mean, so RNA 3D structure prediction in particular is very challenging. I think that for the types of RNA that are common in the PDB, we can already do quite well, but those are usually not the ones very interesting for engineering. So for those kind of more challenging RNA nanostructures and machines that we want to design, I think 3D structure prediction is not there yet, actually. Um, there are people working hard on this. Nature Methods has claimed that 2026 RNA structure prediction will be one of the methods to watch out for. So watch out. Uh, and so, but in my work, like on gRNAde and designing RNA structures, or like a structural basis for RNA design, um, we thought we should try this anyway. Um, and how we actually how we work on design currently is we generally don't use 3D structure prediction to kind of to select the best designs. Instead, we use other sort of models. So for instance, usually you're designing for some function. So if you could train a model to predict function, that could work, right? That's usually very hard, but if you can, that could work. Um, Otherwise, you could use secondary structure prediction, which is that base pairing patterns that I talked about. It's only saying which nucleotides or which positions in the RNA are paired to which other ones. So a simpler task, and we're quite good at this. So the state of secondary structure prediction is a lot better, um, as well as predictions of maybe you could call it like other biochemical structure related properties of RNAs. So in this case, there's this idea of chemical probing or chemical reactivities of RNA molecules, where essentially, at a high level, what you do is you take your RNA and you attack it with some small molecules, and then you're able to measure which portions of the RNA are damaged versus which ones are not. And this gives you hints about RNA structure, because if the structure, if a position is highly structured, if it's part of, say, base pairs, it will not be attacked by the small molecule. Whereas if the position is unstructured, it's quite dynamic or quite flexible, then the small molecule would be able to attack it. So this gives you something like a chemical reactivity profile of an RNA. And we found actually in the gRNAde paper that these reactivity profiles and being able to predict them from sequence is already quite useful for RNA design, can be used to screen RNA designs. So we've been using this sort of workflow where we predict the secondary structures and these biochemical properties of our sequences, and use those to filter down and to select good design candidates.
Yeah. So this is an interesting thing. In the world of proteins, right? Like you may have a model that is good at designing proteins, and you go and say, okay, I want to design a protein with this specific shape, which typically would be rigid somehow, and then you go and get the structure of that protein. So you can go from A to B and then compare, B to B, right? So if you say, this is my sequence, it's going to get this structure, and then I experimentally measure that structure and I see, well, does it match or not, right? Whereas in RNA, because it's a lot harder to get those structures, you can say, okay, we're going to decide RNAs that perform functions, I guess, catalyzing some reactions. And then you want to say you design the RNA and then you measure how the RNA catalyzes that specific reaction, right? So it's a little bit more of an indirect, I guess, but at the end of the day, if you're designing molecules that perform a function and then you can measure the output of that function, right? Like you're, I would say, confident that you're actually doing a good job at it, right?
Yeah.
Like, did I get that right?
Yeah, I think so. I think that in protein design, structure prediction or forming a particular structure is only just a proxy for performing the function you want, right? And molecules which just form a specific structure are statues, and molecules which are dynamic and functional are machines, right? And I think we don't really want to design the statues, right? We want to design these functional machines.
Yeah. Well, sometimes we do want, I guess, just the kind of like the type binding into a virus or whatever, right? Like if you have an antibody, I don't know if you care so much about like how does it move, as long as does it really bind, your epitope or whatever. But again, I'm not an expert either, so.
I think the developability properties depend on how it moves, no?
Fair enough.
I'm also not an expert on the antibody design stuff.
Cool. So maybe going a little bit deeper into the model, right? And the kind of the internals of it, right? So of course there are many different ways that you could have built that model, right? Like processing just sequence, or processing structure, or being accounting for where the relative locations versus absolute locations of atoms are, and so on. Maybe at the higher level, right? Like from what I understand is an equivariant model, right? That is you kind of you start from I guess a notion of like the structure that you want, right? And then it gives you what would be a potential sequence, right? But how has the architecture of that model, right? Like how How did you end up with that one? How do you get your training data and so on? A little bit for like, maybe if a machine learning practitioner wanted to reproduce that, or like, what are the key concepts behind it? And how does that work?
Yeah. So the way I think about the grenade model now in 2025 is take the analogy of prompting an LLM, right? We construct a prompt which contains some constraints or um, something we want to design, and then out comes a sequence of text that um, largely follows that prompt, right? So for RNA design, gRNAde is similarly an RNA language model that can be conditioned or prompted with structure. Uh, so this structure is your constraint or your goal or your target. And this can mean a 3D backbone. So this is a 3D structure without any information about the nucleotides, only the coarse, sort of high level skeleton of a 3D structure. Additionally, your prompt could contain information about the secondary structure, which is, as I said, like the base pairing patterns, which nucleotides are paired to which other nucleotides. And and if you further so want, you could also fix the identities of certain positions. So certain nucleotides, you could say, force this nucleotide to be a G and this other one to be a C, because maybe from biology that this is absolutely essential to preserving function. So essentially, gRNAde allows you to construct this multimodal prompt. This is also done in like protein design as well. And it takes this prompt and it needs to somehow feed it to an RNA language model that's going to generate the sequence that the model thinks is going to, obey these constraints. So how it does so is it takes the prompt and it passes it through a graph neural network, um, and then takes the embeddings from the GNN into a language model. Now, the GNN is a little bit special. It's it's processing what's known as a geometric or 3D graph. So, uh, it's a graph which for which the nodes are objects in 3D space. So these are the nucleotides. And the connectivity of the graph is determined by proximity in 3D space, as well as those secondary structure base pairing patterns that I was talking about. So you kind of construct a graph of, let's say, the 16 nearest other nucleotides, as well as any nucleotides that are paired. So that gives you, this 3D object, which is like the prompt that you gave, but can now be processed by a neural net. Um, and this object, this geometric graph, is fed into a GNN that is specialized for handling such 3D objects. This GNN is, as you pointed out, it's a 3D equivariant graph neural network. Um, and what this essentially means is that it's a model that's equivalent to rotations and translations of your input. It says that if this is my geometric graph or my RNA backbone structure, I can move it around in 3D space, and it's the same. And I can rotate it, and it's the same. So I'm gonna build this property into my neural network, that the model's output is going to transform geometrically. It means that the embeddings that this model produces are actually, geometric objects themselves, and you can apply the same rotation to the input, and your output would rotate covariantly or equivalently or in the same manner. Um, so this class of networks was something I actually spent a lot of time studying in my PhD, and we end up applying it as part of gRNAde here as well. Um, and yeah, this is one way to kind of take a 3D object and encode it in a, let's say, a mathematically principled manner. And then you can use these embeddings as input to an RNA language model, which uh, is quite standard. Like it sort of autoregressively generates your sequence. And as an advantage of that, you can then apply all the sampling techniques to this RNA language model as well. Uh, so this is how it kind of works at a high level. It kind of combines some generative modeling as well as some ideas from geometric deep learning or representation learning to kind of package things together.
Yeah, yeah, which of course, I think makes a lot of sense, right? Intuitively, if you think about the real world, physical, measures in many ways, right? A lot of the world that we can measure, that we can touch, is equivariant in that sense, right? If you move objects, if you rotate them, you're going to have to get the same result, right? So if you have a machine learning model that predicts the weight of an object or the volume, and then you rotate that object, volume is going to be the same. Like we know that from the laws of physics, right? On the other hand, and you would imagine that these equivariant models, give much better results in anything that is related to predicting, real world stuff, right? But then on the other hand, you have sometimes video generation models that can, move objects around, and they have no notion whatsoever of equivariance. They have no notion whatsoever of physics. They don't understand preservation of volumes, right? They don't understand any of that stuff. And then they generate video, for example, that looks hyper realistic. And my understanding too is that there was a little bit of a push a few years ago for equivariant models because again they make so much sense. But then people have made non-equivariant models. I think AlphaFold 3, right, is one of them. They're not equivariant, but they get really good results, right? So maybe where do you fall in that camp, right? Like do we actually need to impose these constraints to have richer models, or is it just a matter of like let's get more data and then the machine will figure it out?
Yeah, I mean, I fall in the camp of now. I fall in the camp of I want to build useful models, and whichever technique empirically performs the best, I would like to use that. Um, so that's the camp I currently fall in. But I've actually written about this in detail on my Substack, and I discuss this a lot. My background is in machine learning and architecture, so I love this discussion. Let me summarize my current thoughts. Uh, I think that for something like RNA structure, where the scale of your data is quite small, um, I think you really see some benefits from equivariance and building this kind of physics into your architectures. Because, you know, what is equivariance or what are these inductive biases? These are also, as you said, physical laws or constraints that we're building in, right? So if you are working in the limited data regime, then your model doesn't have to relearn these constraints while training, right? And so it can be advantageous to use these kind of geometric architectures. Now, when you flip the picture and you're not as limited by the data, you are more limited by, or your performance is more limited by how long you can train the model. Um, and you have a lot of diverse data. I think that practically what happens is that the current generation of equivariant and geometric architectures is computationally weak. It's computationally quite slow. It's quite cumbersome to accelerate them. And at the same time, through the rise of LLMs, companies like Meta and NVIDIA have invested so much brain power into accelerating one block, the transformer block, right? So you can use this acceleration to essentially train for much longer with techniques like data augmentation to learn all those symmetries and essentially have a faster and more flexible architecture. And you really see this, let's call this the hardware lottery of these architectures in play in diffusion models for biology. I think that when you train a diffusion model, then you kind of know that they learn quite slowly compared to most supervised learning models. And thus you need to train them for quite a while relative to supervised learning models. So in the same compute and time budget, using a pure transformer architecture, you could perhaps do 1,000 times more training steps than an equivariant network. And so the kind of nice sort of physics benefits of equivariance are almost washed out by just the duration of training needed to get the diffusion model to work well. And we're seeing this repeat over and over again. We've seen this for proteins, protein design stuff, obviously AlphaFold 3, but also for like small molecules, for crystals, across like biology and chemistry. We're seeing this play out again and again.
Yeah, yeah. I mean, that's the what was the name of the famous paper? The Bitter Truth, right? I think it was
The bitter lesson. Yeah, there's a blog post by Richard Sutton.
Yeah, at the end of the day, if you have more data, that wins. And if you have, a very big differential in performance, right? 1,000x, like you mentioned, you might as well learn the laws of physics again from scratch instead of, running 1,000 times slower, right?
Yeah, I think there's some results. Like I think it's not conclusive yet, but for certain papers, they try to show that if you didn't care about the sort of computational speed aspect of it, the scaling laws for an equivariant architecture might be better than a non-equivariant one. Um, but I think it's hard to argue because the computational speed is currently a lot slower. Of course, in these models.
Yeah, yeah. I mean, at the end of the day, right? Like, what you want is to solve problems in the real world, right? Like, not so much, solve theoretical problems, but the actual problems, right? So, maybe going a little bit back and maybe putting everything together, right? Like, so, what are the kinds of things that you would want to do, if you had, or with your current model, or as as the model improves, like what are the kinds of things that you would like to do? Saying, what kind of ribozymes, would you like to design? What kind of other structures, you would like to create, and what would be the, the maybe practical application? Like, would this be therapeutics? Would this be, synthetic biology constructs, right? Like, what kinds of things would you like to build?
Yeah, I think the answer is all of those. I think but let me try to give you one example that we haven't talked about. Okay, I mean, before I do, yeah, I think I'd be really interested in the ribozymes problem, right? I think they're very fundamentally interesting to biology, and being able to design ribozymes reliably, especially for new functions. So we explored in our paper RNA replication, RNA copying, uh, but maybe for self-replication. Now that would be really exciting and would really push fundamental science forward. Um, but let me give you an example more to the realm of therapeutics. So we know from, say, bacterial mRNAs that they have something known as riboswitches included in mRNA. So mRNA contain a coding region which codes for proteins, and then two flanking regions on the sides which do not code for proteins but contain other sort of elements which are essential to the function of mRNA. Usually these other elements are actually quite structured. So there are regions where the ribosome kind of attaches, and so on. One of the really interesting regions in bacterial mRNA is called riboswitches. These are RNA sensors. So they look for, let's say, some metabolites in the cell. They're essentially detecting where the mRNA is. And when they detect, say, some small molecule, they turn off or on, which means that in the presence or absence of a certain small molecule, your mRNA will start producing or not produce a protein of interest. Or it may regulate the amount of protein it produces. Usually biology is not so exact. It may regulate the amount, right? So this is a really interesting mechanism that is like currently missing from current generation of mRNA and RNA-based therapeutics. But I feel like, wouldn't it be really interesting if we could engineer RNA therapeutics that are truly programmed in this way, that can detect certain cellular conditions, perform some cellular logic, and then activate or deactivate the payload. And I see this sort of riboswitch design problem as moving towards this direction. So if we could, on demand, given a certain small molecule or maybe even some other RNA transcript or another protein, if we could design riboswitches and really like control their sensitivity, how much they turn off or on, I think that could really enable something quite new in the therapeutic realm. And that's like one dream problem that I'm quite interested to work on. And I mean, honestly, like the current grenade and the current set of tools is not enough. I think these new problems also inspire like new methods development. So I think like, we need to think about new methodology as well to really start tackling these sort of programmable RNA or RNA computation design problems. But yeah, this is like the sort of this is one of these dreams that I want to work on.
I mean, it's before going down deeper in it, I was keep trying to think of a metaphor that somebody who's not an expert on biology could understand, because in my mind this is very, very shopping, right? It's like, if we go back to the analogy of like, well, you have your blueprint. Your blueprint is just a piece of paper, and then at some point something builds an object out of that blueprint, right? And what you have here, what you were talking about, is like, well, you have this piece of paper, the blueprint, but then it kind of becomes an origami figure, and then suddenly it becomes a picture frame, right? And then it performs a function, right? It holds a picture, or you fold it in a specific way and becomes a mug, right? And then you put liquid in it, and then it kind of does something else, right? Maybe it opens up a hole or whatever, right? So this I guess RNA molecules not just belong to one of these two functions of like transmitting information versus performing some reaction, right, or enabling some reaction, right? They can do both things, I guess, at the same time, right? So from a engineering perspective is it's very confusing, right? But I guess biology is always full of exceptions anyway, right?
Yeah, yeah. I think like playing on this theme of information coding and functionality, I think that's what is like really interesting about RNA biology.
Mhm. So, like for example, in I guess in bacteria, if you were able to say, have a really good way to understand these methods, you could find a small molecule that somehow inhibits, that riboswitch from working, which
Yes.
prevents that bacteria from doing what it wants to do, right? And then, that could be a great antibiotic, for example.
Yes. This is a very active line of research as well, right? Targeting these bacterial riboswitches. Yes.
Cool. So yeah, that maybe we've kind of, I think kind of, gone full circle, right? Like, so then how did you get started into this space? How did you get interested? Like, what was the path that took you here? As you said, you come from an ML background, right? Like, so what got you interested in space?
Yeah. I think m- contribution of many different factors, as it usually does. One was COVID-19 and wanting to read about what's actually going on in the world. I had left studying biology in high school, but this made me revisit biology, and I thought, like, wow, this is really fascinating when I try to learn about something out of my own interest. unlike, being forced to study it at school. So I really liked reading about the biology. Um, Other factors, well, I think at the same time I was starting my PhD, and I chose a lab where, it wasn't just a machine learning group, it was a computational biology group. So I chose a lab where I'd get to speak with people coming from a different background than myself. And I think that made a huge difference because I could ask them what's exciting and talk to them casually over lunch and ask them dumb questions. And that led to wanting to find something personally meaningful to ground my AI research in. And I think like biology, medicine, like these things, this was something that really stuck out to me. Then specifically finding like a niche in RNA, well, I tried reading textbooks and I found it really hard. So I started to read a lot of popular science books, like biography of scientists, or books by authors like Siddharth Mukherjee about like cancer and cells. Um, and when you do this, it starts becoming clear that, you know, RNAs are what's like really interesting to basic science. Similarly, conversations with biologists. So I was at University of Cambridge, which is a quite small place in that you can interact with people very closely. Talking to biologists there, like I quickly learned that, hey, they're really interested in, processes that involve RNA. Whereas when I looked at the machine learning world, everybody was interested in, say, antibody design or proteins broadly, right? So I thought, this seems like something like forward looking or like an attempt to, explore a new direction. Um, and I think somehow things stick. So I'm still reading about RNA biochemistry. I really enjoy reading it. And, I'm still fascinated by biology and by, this field in general. So yeah, I mean, I entered it through these various influences of my surroundings. And then something stuck, and I just kind of kept working on it. I think that when I started doing this grenade project, it was meant to just be like this side project to explore some new applications of the machine learning models we're building. But it also like caught the attention of some of the top like RNA engineering labs at Cambridge and at Stanford. And I think that made all the difference. Actually, the working with experimentalists was so thrilling, and they, held me to high standards. And some of the molecules actually worked in the lab as well. That's why we went and wrote a paper about it. Um, I think these experiences just ended up propelling me into wanting to, go deeper into this field, move beyond just side projects to, um, really think about like what's the frontier and what's next. Yeah. So that's how I kind of got into this space.
Definitely. Yeah. I mean, I think it's super interesting. And before we talk about like, what's coming into the future, right? I think one of the things that you mentioned was, maybe a few folks interested in antibody design and other problems. I imagine it's because there's a lot of commercial interests, right? Like, some of these antibodies, they are, like functionally, I guess the mechanism of action is reasonably well understood now. And if you could somehow design one really, really good, then there are billions of dollars, right? That you could possibly make, right? I think an interesting thing that you mentioned, right, is working with experimentalists, which of course makes a lot of sense, right? And I feel like maybe one of the reasons why progress in biology has been a little bit slower than it could have been is because there's a cycle that takes a substantial amount of time, right? So, like, you may think about some mechanism that you want to understand better. You design your experiment, you get your, animals, for example, or cells, you run your experiment and you get your results, and, everything is said and done. And it's like 6 months later, and then you go and analyze the data of your experiment. Whereas in machine learning, or in software engineering, you think, oh, actually, this algorithm is faster. You write the algorithm, just run it, and then 10 minutes later, right? Like you have a benchmark, right? So there's a big difference between iterating every 10 minutes or iterating every 6 months, right? Um,
Yeah.
so yeah, I mean, I wonder, maybe before we talk about like what's coming in the future, right? What do you think are maybe some of the things that computer scientists, for example, could do to accelerate progress in biology beyond, just trying to make things faster?
Yeah, I'm thinking about this myself because I'm also new to the lab stuff. I think that, I think there are already attempts to attack this from various angles, right? So lab automation and like attempts at robotics are obviously a big thrust, right? Now, what I saw like at more academic minded labs is that robotics and lab automation works well when you have a repeated industrial like process, right? And you can automate this. But in in the setting of discovery, many times you're doing things that are so new that it's hard to, the recipe is not set, right? If the recipe is not set, you can't automate it. Um, I think the other thing that, we do, um, and there's more and more like, there's more and more attempts at doing is essentially establishing these computational funnels, right? Wherein like instead of experimentally characterizing a large pool, you computationally characterize them and essentially funnel down to what you're going to experimentally do, right? And I think this basic high level idea has been applied to many realms, right? For small molecule screening quite successfully. Um, and say for protein or RNA engineering with directed evolution, you can also sort of use these types of ideas to accelerate it. So I think this kind of putting computational filtering in the loop with expensive and slow wet lab processes, I think this is quite interesting to me. Uh, I think that this is less in vogue or less sexy than like the generative model that like creates a perfect design in one shot. But I feel like this kind of predictive models for filtering are already like making a meaningful impact. Um, I think the third thing is like either collaboration where the computer scientists make the effort to like be physically embedded in the wet lab and understand the work there so that they can know like how specifically to accelerate it, or ease of use of these tools like education or tools such as like what you guys are developing at Polling, I guess, right? Like you can interact with these tools through either tutorials such that the biologists could use them themselves, or maybe like interact with them through new language models, right? I think that can be really powerful actually. in fact like in fact, I mean, it doesn't help me as a computational person to say this, but I've been like, I've been really like excited by the pace of progress in like LLMs and the potential of integrating them with scientific workflows to essentially empower the scientists doing the experiment, right? Um, will we be able to completely cut out the computational person? I mean, maybe again, like maybe for some workflows we can, and maybe for the more complex stuff you need really experts. But I think the potential is there.
Yeah. I have a funny anecdote. Of course, I don't have much experience working with experimentalists, but I do remember visiting one of the bio labs at the University of Washington here in Seattle, and then they were doing experiments on yeast. And then one of the experiments involved some kind of cell division, right? So there was somebody sitting in a microscope looking at the yeast cells and kind of figuring it out when they um, replicate. And they literally had somebody sitting at that microscope, counting the cells, right? And then, people would take shifts and then they would spend hours, just doing that, like entire days. Uh, and I thought, as an engineer, right, a software engineer, it was like, this is absolutely lunatic, right? But I guess there's just they didn't have another way to do that, right? As a software engineer, if you need to copy 3 values on a spreadsheet, the fourth one you're writing macro, right? But as a biologist, it'll be like, yeah, and that's the process. That's the way we've done it for years, right? So there's a lot of value, I guess, in like sitting next to each other, right?
Yeah. Yeah. I mean, I think it's also like it makes you also respect each other's way of working. And
Mhm.
for especially machine learning people, a data set is just something you download from the internet, and maybe you read the metadata, but you mostly just benchmark. uh, for biologists, data set is physical labor, right? Like,
Yeah.
there's so much work that goes into preparing it. Then, like, because you've done the work, then you're also gonna do the work, like you said, of like counting or like statistically analyzing it carefully, being rigorous, right? Like that connection to your data, like when you have that, when you own the data, I think that makes you really careful and rigorous. And I mean, the hope is that that's what leads to good science and new discoveries, right? I think that was something really valuable I personally took away from like being in a wet lab as well.
Yes. So then, what is next, right? Beyond, improving, your accuracy of your model, right? Or whatnot, what do you think is next in, either your work or like, the field as a whole?
Yeah. I think in the field as a whole, I think there are few like really interesting trends that I would be like closely watching in the coming year. Um, I think the one is like how we will integrate LLMs into some of the like bio ML workflows.
Hmm.
There's excitement for this, and I think you can see that all these big labs also have science teams now, right? So I'm really curious to see what comes out of that. I'm curious to use them in like my future projects as well. I think that LLMs have already accelerated my programming, but can they be used for new discovery or accelerating scientific pipelines? Um, I think the the sort of the other trend I'm interested in is currently a lot of the success stories and results we see in protein design, in RNA design, like generally in academic papers in the public realm, are around design campaigns where mostly you do one round of testing. But I'm really interested to see us move beyond this to like iterative improvements. There's a few works on lab in the loop, but I think that's where like the real power of generative models actually lie, is in the fact that like when you get the feedback, then can you improve your next generation of designs? And how far can you push these improvements, right? I think I want to like explore this. I want to see more of this kind of work as well. Um, so I think like these are like two of the big things. Other things, I mean, people people are really interested in both for RNAs and proteins. Like I'm interested in like the RNA side of it, but also the protein side. People are really interested in moving beyond the current paradigm, let's say, which is this kind of paradigm of like RF diffusion or bolts and so on, right? Of generating a structure and then maybe like generating a sequence for that, doing motif scaffolding, maybe given some functional motifs. Um, so I think like if we want to design, say, multi-state or dynamic molecules, do we need like a different approach to this? So I think this might be something that a lot of people, attack with different techniques, like the problem of multi-state design generally, and the sort of the transformation of the field from static structure-based design to truly multi-state sort of control over functionality.
Massive problem, right? Like,
Yeah.
definitely in the protein space, but even in biology, right? Like, many
Yeah.
things done.
I mean, everything is multi-state in biology. Like when you think about it, right? Like it's the sort of the structural biologist view is, and maybe I can link this to something else I'm thinking about. The structural biologist's view is we're gonna look at a dynamic biological process in one frozen state, and that's gonna teach us a lot about that process, right? And this view has worked in designing new biomolecules as well. It's a rational or mechanistic basis to design, right? But all these structures from the PDB, they've been mostly curated and collected for human understanding, right? And now we're saying we want to train AI systems on the same data. Maybe something more forward looking is maybe the AI systems do not need like the same kind of mechanistic basis to understanding functional biology. Maybe we can also explore an alternative black box approach to kind of using very high throughput functional data collection. Maybe only linking sequence to function, for instance, bypassing structure, but doing this at an unprecedented scale. Maybe that way, maybe this kind of black box data is better for training AI systems. This is an empirical question, so I also want to find out. But I think this is a promising bet. So far, science has generated data for human understanding. Now we're saying we want to generate data specifically for training better AI models, right? This is different. This is new and potentially exciting.
Yeah, I mean, I think that's a super interesting question. Could it be possible to somehow generate, synthetic uh, structural data of like proteins moving, right? And then somehow the AI can figure out, how things work better than humans ever could, right? I don't know, like maybe same as like could you train a machine learning model on video, and then suddenly that model knows how to create images and text and explain a story, right? I don't know. Like people have tried different things like that, and sometimes it works. You just don't know, right?
Yeah. Yeah. I mean, I think we have to try it, right? And I think there are people trying, there are people betting on this sort of black box functional data approach. I think it's really interesting. And it's also like one of the things I'm quite interested to explore because, especially for RNA molecules, these are extremely amenable to sequencing, right? Next gen sequencing. So if you can set up your functional experiment, your functional assay as something based on sequencing, you can collect functional data on like millions of variants potentially, right? This is very powerful. If this is very powerful for iteration. So yeah, this kind of like high throughput data generation capabilities, I think this would be something really exciting to combine with generative models.
Yep. Um, I will have another conversation in a year, and then we can catch up on like what actually came up, and some of the people that are trying some of these things. But this was I mean, super interesting conversation. Thank you so much for taking the time. We're already an hour and 5 minutes in. So
Thanks for your time. Yeah.
no, thank you. This is super interesting. if somebody wants to, connect, ask questions, reach out, maybe what is the best place to connect? And
Yeah, I'm my email and like my profile is all public. All my website is chatjo.com, and my Twitter handle is also at chatjo. So you can find me on various online platforms. Happy to connect with people about all these topics that we talked about today.
awesome. I will, yes, I'll try to remember to add the links at the bottom of the video as well for everybody to reach out. Thank you again. Super interesting conversation. I enjoyed it very, very much. And I think it was super instructive.
Thank you. Likewise, Javier.